in an intriguing new twist in regard to the human aging brain. And please, I'm emphasizing the word human, because a difference was discovered in regard to at least an aging brain when looking at rodents and non-human primates. We'll get more into that in a second. But to start with this particular study, which again, how to describe, reveals more about the human brain that we did not know than we actually do know. But to proceed, older adults grow just as many new brain cells as young people, a deductive statement. There has been controversy over whether adult humans grow new neurons, and some research has previously suggested that the adult brain was hardwired and that adults did not grow new neurons. The study appeared in the Journal of Stem Cell on April 5th, and it counters that notion. The researchers state the findings may suggest that many senior citizens remain more cognitively and emotionally intact than commonly believed. To quote the researchers, "We found that older people have similar ability to make thousands of hippocampal new neurons from progenitor cells as young people do. We also found the equivalent volumes of the hippocampus, a brain structure used for emotion and cognition across ages. Nevertheless, older individuals had less vascularization and maybe less ability of new neurons to make connections." Meaning, the brain basically can produce what it needs to produce. However, if other health factors are involved in the brain, it can hamper that effect, or benefit, I should say. In rodents and primates, this is important, and I'm going to emphasize this one particular paragraph out of the research because so much research is done on the brains of rodents and primates. So, it's important for other confounding or potential confounding in the future that maybe a lot of this old research needs to be looked at again. And you'll understand more why in a second. Let us proceed. The ability to generate new hippocampal cells declines with age. So, in rodents and primates, the ability to generate new hippocampal cells declines with age. Waning production of neurons and overall shrinking of the dentate gyrus, part of the hippocampus, thought to help form new episodic memories, was believed to occur in aging in humans as well. Now keep in mind, I'm going to post, not right this second, but towards the end, a main segment of the full published study for those researchers that want to glare at it a little bit, or should glance at it, and to see exactly what their observations were. But to proceed, the researchers from Columbia University and New York State Psychiatric Institute found that even the oldest brains they studied produced new brain cells. Quote, "We found similar numbers of intermediate neuronal progenitors and thousands of immature neurons," they wrote. "Nevertheless, older individuals form fewer new blood vessels with brain structures and possess a smaller pool of progenitor cells, descendants of stem cells that are more constrained in their capacity to differentiate and self-renew." I'm going to go, before I post the last article I'd like you to review, the conclusion of the full published study, which is not obviously in the public release, states as follows. Thus, healthy older subjects without cognitive impairment, neuropsychiatric disease, or treatment display preserved neurogenesis. It is possible that ongoing hippocampal and neurogenesis sustains human-specific cognitive function throughout life, and that declines may be linked to compromised cognitive emotional resilience. All right, the DOI will be stated so you can follow on your own, but I'll leave this one section up from the full published study. Old non-human primates and rodents have more GN being granular neurons. So basically, as you read through that, that's what the GN states. But that's up there. Pause it, review it on your own. After that, the UI is listed. Follow up on your own as well. Again, this is Ralph Turchiano. Hope you find this information of use. Incredible new discovery. And thank you very much for listening, as always. See you next week. Bye.